Welcome everybody. It is my absolute pleasure to introduce Jajit Das, who is also my college mate. So we did our BSc together. So Jajit did his PhD from math science with Madan Rao, and after that he moved from completing his PhD in 2000, he moved to Virginia Tech and did postdocs in Berkeley and MIT. So that's when he moved from Heisenberg spin chains to whatever more biologically motivated problems. And I myself am very curious to learn what he has been doing. This is very new to me, and so I hope I'll get to learn. And please feel free to ask questions. This is not that he's going to give all of his research today. Is basically to introduce us on what immune systems he's working on and how faces can look at it. Okay, so it will be in the colloquium mode, and I will hand it over to Jayjit so that he can introduce us to his work. I'm very curious. Thanks, Jayjit. Please take over.
Okay, thank you, Apratim. It's a great pleasure, and thank you so much for the opportunity. Um, as Apratim mentioned, I'm trained as a physicist, and I started working in this area about probably 15 years ago or so. And when I started, um, apart from learning the jargons and getting used to sentences like when a T cell receptor is engaged, um, what also amused me and somewhat excited me also, that how flexible the nature of the problem can be in immunology. So what is clear, that it's clear what I want to explain, but what is not clear, what can I do to explain it? So unlike physics, where modeling is very constrained and guided by principles, here there are no set of hardwired principles, and the modeling can be very flexible. You can think of many different models to explain it, and I guess the final test comes from performing more experiments. So it's a different type of modeling and the way of looking at problems, and it's probably similar to the times of Kepler, when there was very few guiding principles. You had this brilliant data before you, and you were trying to explain it by different in different ways. And to me, if trying out different models, if one or two stick, that's extremely rewarding. Um, what I'm going to do this evening, a morning for me, is to talk about two examples where we have tried out uh, some modeling to explain two immunological problems. And I will also set up the problems by giving out details that are only required to set up the problem. So feel free to interrupt me anytime and ask questions. So, oh boy, how do I change slide? Oh, oh boy. Okay. Sorry, I was not able to change the slide. Okay, so now I guess in this times, this picture is enough to impress upon you that it's a multi-scale problem. Our immune response is a multi-scale response. So a virus could be a very familiar virus to us. It could be, it's about 100 nanometer in length. And if you look at the timescale for conformations of the proteins on the surface of this virus, it's about a millisecond. And then this virus gets inside our body, and this micron scale protein molecules start interacting with protein molecules in our body. And then they generate a response that spreads all over our body and causes responses that can last for days to weeks, right? So if you look at the change in length and time scales here, it's about 10 to the power 7, more than a million, and then for time it is even more. So it's clearly a multi-scale response. Now, what I'd like to describe here, that this multi-scale response also occurs in multi-dimensions. So if we peel a layer, if we go a layer deep inside the description I provided, we get this following picture, that when a virus enters our body, it's cut up by cells of our immune system into small peptide fragments. And those peptide fragments are sensed by protein molecules that are present on the immune cells. And this interaction, this peptide came from the virus, and this protein is on the immune, on one of our immune cell, and this interaction is happening in nanometers. And then these immune cells, which are part of our innate immune system, responds to it. So there are two parts to our immune response. One is the innate immunity, which is the first line of defense. So these are the cells which responds immediately when a virus gets in. And so the response starts in seconds and lasts for days. So these cells become stimulated or excited, and it secrete cytokines and chemokines, which are basically molecules that go and alert the next layer of immunity, which is known as the adaptive immunity. So why adaptive? Because this part of the immunity designs itself so that it can go and target the infection that is there. So every infection is different from each other, and the adaptive immunity recognizes that and designs a set of arms that are able to go and deal with that particular type of infection. So this second line of defense, or adaptive immunity, starts after 2 or 3 days, and then ultimately both the innate and the adaptive immunity work together to get rid of the infection. So so this is the reason that after 4 or 5 days, if you get a regular flu or something, you start feeling better, because your adaptive immunity starts working, and then they finally get rid of the virus. So now, as I mentioned, that there are this huge change in length and time scales, and if you look closely in the processes that occur within these time scales, you will find many of them are synergistic, which means that the sum of the parts are, so the effect of the parts is more than the sum of the parts. And this there are many, the synergistic processes occur at all these different length scales. It's very common to find such processes. Now, similar change in scales is commonly dealt with in physical systems. I'll come to that in the next few slides. For example, formation of snowflakes from water vapor, and you can come up with your favorite example, and you will see, and these are the type of things you do. So tools from statistical physics or other disciplines of physics can be very useful. Now, what I'm going to focus on here is that the unique aspect, this multidimensional aspect of our immune response. And this is the part which is not very common in physics. So if you look at an immune cell, you will find that there are about 3,000 different proteins, and all of them work together to generate a response. And then one type of immune cell can produce phenotypically different cells. That means those cells can be functionally different. And the type of different cells, the number of different cells you can create in this process could be well over 6,000 or more. So this is a big number, right? Even for innate immunity, if you consider innate immunity, you will get a number of 6,000, which is a huge number. And if you go to a cell in adaptive immunity, the number of such different types of cell that can be produced by a single cell could be well over 1 million. So there's a huge diversity that one has to think about when one tries to understand the immune response. And this diversity in the response is this multi-dimensions. And this is usually not encountered in physics. And that's why we need to adapt tools from physics to deal with this, because you know, you start any problem and then you will encounter this diversity.
So, Jajit,
Yeah, go
you
ahead.
mentioned that 6,000 kind of cells, they make additional kinds of cells, or did you mean proteins? I mean, each of these 6,000 make proteins, and so you get 10 to the power 7.
Ah, so uh, okay, so let me, so there are two parts to this question. So the first part refers to the innate immune system. And so when I talked about different types of proteins, every single cell will have, let's say, 3,000 different proteins. And if you have protein A, let's say in large number, this cell will behave differently than a cell which has large number of protein B. But
Okay.
both these cells will have protein A and protein B, but in large or small numbers. So because of these different combinations of protein A and protein B you can have in this cell, you can create different versions of the same cell, which will work differently, right?
And then there are 3,000 kinds of cells, and each cell can come in multiple versions.
Multiple version, like
Okay, okay.
when you are teaching, right? You put on a teacher's hat, right? But when
Uh,
you go in the kitchen, you are a cook, right? But it's
right.
you.
I see.
So
I see.
okay.
Yeah.
uh, and then the 10 to the 7 is more interesting. So so this is related to adaptive immunity, and every cell in adaptive immunity has a unique set of receptor proteins, and the number of this unique number of receptor proteins can be over 10 to the 7. So we'll have those cells in our body, but the number of those cells are very small. Maybe you have one type of cell present in one or two numbers, another type of cell present in one or two numbers. But when an infection arrives, a particular type of cell would go and expand
Okay, the
and
population
go
will
and remove the
expand. I see.
infection.
So
So so
these
so
cells
this
are
this
in what,
hmm,
in our bloodstream? I mean, these cells which
yeah,
are small in number, which you said are small in number. I
right,
mean, they are in a particular organ or bloodstream, or all over.
yeah,
Because
okay,
you said it's
good.
a very
So,
low percentage. You said, right, so it has to quickly reach the destination. Yeah.
yes, that really, so that's a very interesting question, right? So how does it happen? So it's like finding a needle in the haystack, and that um, in fact there quite a bit of modeling to explore that. How does the immune system do that? And now coming back to your question about blood and why do these cells reside? So these cells are usually generated from our bone marrow, and then they go through the thymus with some selection, and you will find these cells in the lymph node. So the cells I mentioned, like T cells or B cells, they're found in the lymph node, and some of those cells also go to our blood cells, go to our bloodstream, and the recognition of this foreign bodies can happen at the lymph nodes, where so basically this adaptive immune cells, they recognize and multiply and so on. So to answer your question in short, you will find these immune cells in your lymphatic system, which is shown here, different organs like spleen, liver, thymus, and some in the bloodstream. Does it answer your question, Apratim?
Yes. 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 Please.
Okay. Uh, sorry. Okay. So I guess what I wanted to impress upon you here, that this immune response is a multi-scale response in multi dimensions. Now, in physics, we deal with similar systems, probably not with diversity so much, but um, even a very simple physics problem will involve explaining a complicated behavior by building a simple model where the components of the model would interact. And it's our job, our goal is to find out how those interactions can create complicated behavior like phase transitions in Ising model and critical phenomena. Um, and in order to do that, we use various techniques that are easily generalizable. Right? So if you use a system for Ising model, you can use it to explain other material science system. You can use it for polymers, for membranes. So tools like multi-color simulation, master equation, perturbation techniques, mean field techniques. We construct ODs, PDs, and we have methods for disorder. So all these methods can be used to think about problems in immunology. Um, so in this talk, I will provide some two examples where I probably it will touch on the first 2 points. And the last point is to create simple models that can capture complex and emergent behaviors. So this type of analysis that we do in physics and for Ising model or growth models can be probably used to think about theories in our immune system. So I will not really talk about this part here at all. So the two projects I will talk about here. One has to uh, deal with so one pertains to B cell signaling and how this signaling requires receptor clustering, feedbacks, and synergy between different molecules. And so, and the second problem would be to it's going to a different scale. I will talk about a viral infection in an animal model, and the role of synergy is not that clear cut here, but there is some underlying synergy that will be there. But so I decided to talk about it because of the relevance of this problem in the present times. And in both the projects, we will pursue mechanistic and reduced modeling. So there is a part in the group where we carry out data-driven models, which I will not discuss here. And so this is how we work on projects in our group. So initially, the problem starts with some experimental observation. We analyze the data. We create models to explain that experiment, and then use that model to generate predictions, which needs to be tested by experiment. So this is a very crucial part in our project, that the model predictions should be tested by experiments, and if we fail, we go back to the drawing board and work on the model again. And in order to do so, we need to merge techniques from various disciplines like mathematical sciences, engineering, and data science, and combine them with the tools, with the experiments in medical and life sciences. So the first project I will talk about deals with B cells, and B cells they get the name because they're generated from our bone marrow. So B cell, and these cells are part of our adaptive immune system. And they can be found in the lymph, and they generate responses against viral infections and tumors. So understanding how these B cells work are important for developing vaccines and therapies against cancer. So now, as I probably mentioned briefly before, that these immune cells have receptors embedded in the plasma membrane. So B cells are about 10 nanometers in diameter, and they have B cell receptors that are present in the cell membrane. And these B cell receptors are about 10 nanometers long. There are about 100,000 B cell receptors in every B cell. And the unique feature of the B cell receptors is that every B cell will have a different set of B cell receptors. So in the sense that the protein composition of the B cell receptors will be different from B cell to B cell. And these B cell receptors sense ligands, which are antigens, and this sensing means it can bind to it. It can bind to it with some lifetime. And this binding or interaction is very sensitive, meaning if there is a ligand, let's say this B cell receptor binds to it with a high affinity, let's say it stays bound for 1 minute to 10 minutes or even longer. If you change one amino acid in this ligand, it can destroy the interaction. And then this ligand will not bind to it at all. So this interaction is very specific, and that's the advantage of adaptive immunity, because it can sense the sequences that are really different from our that come from viruses, which are different from the protein sequences you will find in our body, and it will create a response which is designed only for that particular uh, peptide and particular antibody, and not for anything else. So, and to give you a sense of the length scales here, so if you think yourself as a B cell receptors, and you take a stroll in the Central Park, this upper and lower side of the east and west side of Manhattan and the Central Park would be your B cell surface, and this would be you taking a stroll here. So this kind of gives you a scale of how this receptors move around in this B cell surface, and there are about 100,000 of you walking around in this area. So one thing many experiments noticed is that when they try to look at the B cell receptors, they find that these receptors are clustered. So so this cartoon is basically showing the B cell clusters. So here the BCR is a monomer. Now this is a B cell, right? This is a B cell, and this is a lipid layer which are presented with different ligands. And you can see that when these ligands are present, which can bind to the BCR, you form this large clusters of BCRs. So the question we wanted to ask is, what is the functional roles of this cluster? So it's clear that these BCRs form clusters, but why do they do that?
Oh, 1 second, Jayjeet. So
Yeah,
you said they form clusters after they are sensed by a ligand. I mean, they are not naturally in a cluster form. Only if they get attached to some ligand, some outside, then they form clusters. Is that correct? Is my understanding
very good
correct?
question. Yeah. So this picture is when there is no ligand. And then you will see that there are some clustering. So there are some multimers, trimers, dimers, but the sizes of these clusters are smaller.
Okay.
When
Okay.
you present the ligand, some of these clusters become bigger. So
Okay.
there is some evidence of pre-clustering, and there are various biophysical models exploring this, that why this, how this clustering happens.
Okay. And one just another clarification.
Yeah,
You already mentioned that each cell has whatever 1 lakh of these sensors, BCRs,
right,
and different cells have different proteins. So if a different a different infection or a different virus comes, it will get attached to another version of the same cell. Is that a correct
correct,
statement?
correct. Yep.
Perfect. So I've understood it correctly. Thanks. Go on.
Yeah. Okay. Thanks. Okay. So So when you started thinking about that, how BCR clustering is going to affect um, uh, the B cell signaling and activation, uh, this is the picture we got. Now, you don't have to really look at this thing carefully. It's just going to, just to tell you that when this interaction happens, that means when this BCR binds to this antigen, there are various proteins in the cell membrane and in the cytosol that start reacting. And a product of many of these reactions is to activate some genes in the nucleus of this B cell, which would generate proteins which will come out, let's say, neutralizing antibodies. And then this antibodies will come out and go and bind to the, let's say, viral particle, not viral, but other cells that are presenting this antigen, and neutralize those antigens. So this is the type of response one is thinking about when a B cell gets stimulated.
So in
So
what way is the
yeah,
clustering helping? Because this seems like a, I mean, I'm not sure what is the role of clustering of these BCRs.
great. Yeah, that's what we want to find out.
Oh, I see.
It probably will become clearer in the next few slides.
Okay, thank you.
Okay. Um, yeah. So, but what is known that the initial reaction, that the first few reactions play a key role to decide whether further reactions will proceed or it will just stop there. So we decided to look at the first few reactions in great detail. Okay, so this is what I just mentioned. So these are the early signaling events. When a ligand binds to a BCR, attached to the BCR are these cytoplasmic tails. So these are like polymeric chains. And then these polymeric chains have a particular motif of amino acids, particular pattern of amino acids that are present. And in that pattern, there are tyrosine molecules. And these tyrosines, once they're phosphorylated by kinases or enzymes that are in the cytosol and in the plasma membrane. When they're phosphorylated, they can initiate more reactions. So this motif here can be either partially phosphorylated or fully phosphorylated. And depending on whether it's partially or fully phosphorylated, it can start different types of reactions. Now, there are two different types of enzymes or kinases that carry out these phosphorylations. One is Src family kinase that lives in the plasma membrane, which can go and phosphorylate these tyrosines. There is another kinase, another enzyme present in the cytosol that can bind to this phosphorylated tyrosines. And when they're bound to it, it has a kinase domain which can go and phosphorylate other tyrosines in the vicinity. So we started wondering that why do you need two enzymes to do the same job, right? Both the Src kinase that is in the plasma membrane and the Syk kinase that is in the cytosol, they do the same job, right? But why do you need both of them? Is it just a redundancy or has functional implications? And then it turned out it is tied to the clustering question we were thinking about. So this project was led by a very talented postdoc in our group. Now he's a scientist in Battle Memorial. It's Shahid Mukherjee. And we collaborated with our experimental colleagues at UCSF, Berkeley, and MIT to answer this question. So we can now look at how this activation, how this phosphorylation happens via Src or Syk kinase, right? So let's say this is a BCR, it binds to the ligand, and then Src kinase, they're in the plasma membrane, they would go and activate these kinase, right? Now, Syk kinase, on the other hand, it lives in the cytosol. It has two stickers with which it can bind to this phosphorylated tyrosines. If there is only one phosphorylated tyrosine, it uses one sticker, and the binding is of low affinity. If it has two stickers, two phosphorylated domains, it uses two stickers, and so the affinity is high. And when it is bound to this motif, it can go and activate neighboring tyrosines. Now, notice that this molecule, which can be bound very weakly, it can quickly go and phosphorylate this guy, right? And then this binding can become strong affinity. And then it will go and activate more BCRs, which will create more binding sites for the Syk. So it creates its own activation. So there's a positive feedback in there. And now we can start to see that how spatial clustering would enhance this effect. Because if you have neighboring BCRs present, this Syk-mediated phosphorylation would be favored by that. So, but does it really happen? So in order to explore that, we created a very simple model. In our model, we had BCRs, executing those reactions with Syk that I mentioned, and the reactions with Src kinase that also I described. And then there are these phosphatase molecules, which are enzymes, which removes the phosphate tag from these tyrosines. So this is kind of a deactivating enzyme. So, so what we wanted to explore how this feedback is affected by spatial clustering. And I think I already mentioned this activity in words. So, the Syk is recruited, and once it's recruited, it can activate the neighboring guys. Okay, there is another thing I did not mention, that the Syk, once it's bound to this tyrosine domains, it can go to a state where it can execute a greater enzymatic activity. The rate at which it can phosphorylate the neighbors can become stronger. So this is that autophosphorylation, and this also has a positive feedback. So far, so good. Any question at this point? No? Okay. All right. So I think I should probably move faster. Okay. So we created a reaction diffusion model. So the model included all these reactions, and it also had diffusion of the receptors, the ligands, and the enzymes that I mentioned. So we did a simulation where we took a simulation box, which is of size of 5 microns by 5 microns. And this box is divided into small chambers, I think of size 1 micron cube. So in the simulation, the the molecules could hop from one subvolume, one chamber to the other chamber, and the reactions only happened in the same chamber. So if you have molecules in two chambers, two neighboring chambers, they don't react, but the molecules in the same chamber, they react with each other. And this set of events were modeled as stochastic events, and the simulation were carried out with spatial Gillespie method. Now, I guess the crucial question is the rates, right? The rates at which these reactions happen. And most of these rates are not known. So, but some of them can be estimated, from the timescales of activation that you see in experiments, and many of the rates are not really need to be in precisely estimated to give you the expected behavior. So you can vary many of the rates quite a bit and still get the behavior, still get the qualitative behavior out of the model. And we used a package to carry out the simulations where this is freely available. So you can take these reactions and write those. You really don't have to write the Monte Carlo code. So this compiler generates it for you. And but you have to, of course, supply the reactions that you want to model and the diffusion.
So finally, you have a hopping process for each of these six and SRCs and so on and so forth. Is that correct on a lattice? Or are you basically writing down differential equations and discretizing them on a lattice?
Good question. Yeah. So the first question, not every molecule is diffusing. So if this BCR is bound to a ligand,
Okay.
then this molecule is immobile here.
Correct.
And then if other molecules come and bind to it, they're not diffusing. The entire complex is not diffusing. The free molecules are diffusing.
Okay.
Now for the second question, we are using that as events. So these are purely stochastic simulations. We are not discretizing ODs here or PDs here.
Okay. Okay. So you have lattice, and then you put particles at a certain concentration. You make them hop around from one lattice point to the other. Okay, I get it.
Right. Essentially, you are solving this stochastic master equation here using Gillespie's method.
Okay.
Okay, so we looked at two situations. We looked at a situation where this BCR molecules are clustered at the center, is centered like that. And then we looked at a case where these BCRs can just freely float around. And what we measured is the number of fully phosphorylated tyrosines that you can get, because that's a measure of activation. If you have this fully phosphorylated tyrosines, that means the next reactions can occur with a higher intensity. And these are the results. So here in black, I'm showing you the number of fully phosphorylated items. Those motifs are called items, and how their number increases with time. And this is the case when we remove the Src kinase, the kinase that are present in the plasma membrane. And you can see that there is a small drop in the phosphorylation. And now this is for the case where there was a central cluster. In the second part, there was no cluster. They all homogeneously traveled around in the simulation box. And if you perform the same test, same experiment simulation here, you remove the Src kinase, you will see a large drop in the phosphorylation, right? And this is because when it was clustered, most of the phosphorylation here was carried out by the sick molecule via this feedback, because it would find other molecules in the vicinity to phosphorylate. But when it was homogeneous, the sick did not have neighbors to phosphorylate, right? So most of the phosphorylation was done by Src. So when you took out Src, it dropped, right? So fine, so far so good. But now you have to test it, right? Because as I mentioned, there are many ways you can model the same thing. And there are many details I also did not tell you, and that also brings in quite a bit of flexibility when you start modeling things. Okay, so these experiments were done in Arthur Wise's lab at UCSF. So he designed, or his lab designed, a very clever experiment. So they had this antigens, they had this monomeric antigens. And to induce clustering, they created this dimeric antigens. So these antigens now are cross-linked. So when they will come in contact with the B cell, it will also cluster with B cell receptors. And then they use two inhibitors. So there is a drug. If you put in this drug PP2, it will block the SERT kinase, meaning it will, in the presence of this drug, this kinase will lose its activity. And then you have another drug which will inhibit SYK, that it will lose its kinase activity, right? So with these tools, one is able to probe the prediction. So what I'm showing you here is the amount of calcium that is released. Now it's difficult to probe directly the number of fully phosphorylated items. But what happens when you have fully phosphorylated items, it can generate a strong calcium pulse. So with monomers here that are shown in green, you will see a strong, at this arrow, they added this antigen, and you see the strong calcium pulse. This x-axis is time. When you add PP2, that would block the SERT kinase, you will see a dramatic drop. And you increase the dose, you will see the drop happening even more. Oops, I think I went out of the. You will see the drop happening more. Now, he looked at the case, or they looked at the case when the antigens were present in dimers. Again, you see this strong pulse. But when you add the SERT kinase inhibitor, when you block SYK, the drop is not that dramatic. This is consistent with this. And you increase the dose, even the drop is not as much as what you see here. But then you add the SYK inhibitor, and then you see all the activation going away. So basically, most of the activation here for the blue line was done by SYK, as we predicted from our simulations. So, and then there are other tests which you can look at in this paper. So essentially, this clustering helps to create more signaling via this spatial feedback that is induced by this SYK. And so this can come into play when you have multimeric ligands in the system, and this SYK mediated positive feedback that I mentioned can also create binary activation, meaning you can have an all or none response in the item phosphorylation, and that will create a kind of a digital response that if you increase the dose over a threshold, only then a lot of your B cells will be activated. So it creates kind of a sharp activation threshold for B cell activation. So I'm not sure how much time I have, so I'll go through this extremely quickly. Okay.
You have 15 minutes.
Yeah. Okay. So this is a very recent work, and this came about from some experiments carried out by Mark Baith and Daryl Irvine's lab at MIT. So they were trying to create this antigens, or these are vaccine candidates, where they will attach this ligands, the BCR ligands, on DNA ligands, on DNA molecules, right? So you can have a DNA mesh like this, DNA nanoparticle like this, and they would put this this antigens here, or you can have a rod-like DNA, and they will put these two antigens here. The idea is to introduce these nanoparticles in someone and vaccinate them, right? So the idea is that this ligand should go and activate your B cell and generate a response that will help you to fight other viral infections. Now, what they found is kind of an intriguing observation. They found that if they take these nanorods and they have two antigens, and then they increase the spacing between the antigens, right? Now we are talking about nanometer scale. So 1 nanometer to 7 nanometer to 14 and 21 to 80 nanometers. they will see an increase in calcium signaling. The calcium signaling I mentioned. So how changing the spacing between these two antigens is leading to more activation, right? You'd imagine that if they're clustered more, they would produce more. So it would kind of go down instead of going up. So that was an intriguing observation. And then we tried to explain it in the following way. So in the BCR, in the B cell, as I mentioned, they're clustered, right? So these B cells can accommodate, let's say, 5 molecules. And then you have these antigens which are connecting these two BCR clusters, right? Every BCR cluster has 5. And the size of this cluster is about 30 nanometers. Now, as you decrease the size of the spacer, these two clusters start merging. And as they merge, they will exclude BCRs because of steric interactions, right? So for smaller distances, you will have fewer number of BCRs to activate than the case when this antigens were farther apart. And this reduction in the number is essentially behind this reduction of activation as we are decreasing the distance. So that was at the heart of the hypothesis we put forward. And then the sick feedback came into play in the following form, that this antigen here presented by that DNA rod will bring in sick, and the sick would go and phosphorylate its neighboring BCRs, even though they are not bound to any ligand. And this would lead to fully phosphorylation of these items and produce more calcium. And then, our model is able to produce that behavior. And then, there are other agreements that we tried, experiments in Darrell and Mark's labs, and you can read about it in this manuscript. So because of the short time, I'll probably not talk about the future directions and so on. Let me see how much time. So 12 minutes, right, Apratim?
Yeah.
Okay, so I'll talk about a very different problem now, right? So I'll talk about a viral infection by a virus known as respiratory syncytial virus, or RSV. I'm not sure how many of you have heard about this virus, but this is a very common infection that you will find in children. So almost all of us are infected by it by the age of 2. And for toddlers and babies, they can cause serious health issues. And it can also cause serious effects for older people. So it affects people at the extremities of their lives. And they become fatal because they are synergistic with bacterial infection. So you're probably, if you have a small child, you're probably familiar with ear infections. And these are caused by bacterial infection, and which come about after an RSV infection. So there's this synergy between this bacterial infection and this infection by RSV. There are no vaccines. And one reason we got interested in this, because we wanted to think about middle ear infection in children. So the question we wanted to ask here is what are the roles of innate and adaptive immunity in controlling this viral infection? Now, this type of questions have been addressed by modelers for many decades. And one of the pioneers in this area is Alan Perelson, who looked at HIV infection using this type of modeling, and that essentially led to development of cocktail therapy. So it has tremendous therapeutic applications. So the type of modeling here is really simple. So there are these target cells or cells in our body that get infected by viruses. And these infected cells can die, or they die, just, these viruses just go and burst the cell, or they can die because the immune response would come and eliminate the cell. So there is a death. And then, but these infected cells create more viruses, and those viruses go and infect more cells, and the viruses are also cleared in the system. And this model can be described by a very simple set of ODEs. So T, for example, is the number of target cells, and they are infected with a rate beta, and V is the number of viral particles. And this is the rate at which infected cells are created, right? So this produces infected cells, and the infected cells die, right? And infected cells produce viral particles, and the viral particles are also cleared. So the way this modeling is compared with experiments is they solve these ODs, and in experiments you can measure the amount of viral titer you would find in a sample. So for example, Perelson and colleagues are looking at viral titers that they collected from human donors, human volunteers. So these human volunteers were infected with flu, and then you take some nasal swab, calculate, find out the viral titer at different times during the infection. It's going from 0 days to 7 days, right? And then from your model, you can find out what is your viral kinetics, right? You don't have much idea about the T and I in experiments, but you can fit the viral kinetics, and thereby you can estimate some of the parameters here. And from those parameters, you can calculate various quantities like R0. I think many of you are familiar with R0 now. It tells you how many new infections, how new target cells are infected by a single infected cell. So if R0 is less than 1, the infection dies on its own. If it is greater than 1, the infection spreads. So we thought one can probably do this type of modeling for RSV infection. So we started talking to our colleagues, virologic colleagues at the Children's Hospital and Ohio State, Mark Peoples and Stephen Niswick. And this project was carried out by Darren Wethington. He's a graduate student in the group. And we also collaborated with a statistician at our center, Bill Stewart, and we also worked with Alan Perelson. So what Mark and Stephen pointed us to, that you have quite a bit of data available in the literature from very early experiments by Robert Chunnock and George Prince. So what they did is they took cotton rats, which is the model animal. They injected some virus in the cotton rats at different ages. Remember, age is a big factor here because we are also infected by RSV, but unless we are over 65, it's not really a serious problem for us. And it's also, but it's a serious problem also when you are a toddler. So they took cotton rats at different ages and looked at the viral titer in the lungs of the cotton rats, and you get this different viral trajectory, right? Titer kinetics. Now, so far so good. And when it started to think about this, we said, okay, why not see how does it compare with the data you have for flu, right? So if you look at the flu kinetic data, you will find that the decay here is slower, but the decay here from the peak is much faster, right? So the question was, can you use the same equation that Perelson and colleagues used to describe this flu data? Maybe it's possible to do that because you can change parameters and get whatever behavior you want, right? So in order to test that, we thought maybe we should characterize this kinetics a little more. Let's try to see what is the shape of this kinetics. So you can find out the peak time, you can find out the time scale for the decay, and you can also find out, how long it took to go to the peak, right? So you can create a threshold here where the viral titer drops f times from the peak, and then you can get this time scales out, right? And we created a dimensionless ratio parameter, which is the ratio of the decay and the product, time scale for decay and time scale for product. So if it is less than 1, that means you have a shorter decay time compared to the production time. And we calculated this ratio for the data we got from that paper for the cotton rats of age 3 days, 14 days, 28 days, and adult. All of them showed this ratio to be less than 1. So we thought, okay, so let's try to see if we can get this ratio to be less than 1 for the model, the flu model. And we varied the parameters, 1,000 times up, 1,000 times down, to see if it can really produce something at a less than 1 value. And to our surprise, we found that no matter what you do to these parameters, you cannot cross this ratio below 1. Okay? So it told us that this model is not complete. It would not be able to explain the RSV kinetic data. So we went back and modified the model. We started thinking about the problem. So for flu, one of the way the infected cells die is by natural death. But for RSV, what our colleagues mentioned, that the immune system, the adaptive immune system, comes into play, and it plays a major role. So maybe one should include that. Maybe we should put in a large decay of the infected cells after 3 days or something, when the adaptive immune response comes into play. So we modified this equation for the infected cell by adding one term where a strong decay happens after a time scale of 4 days or so. And we added the term and looked at if it is able to capture the ratio. It is indeed able to do that, right? So that gave us confidence that, okay, we are able to test this. We are able to use this model to describe the data. And then, we could fit the data. I will really not go into the detail because of time. We could vary parameters and do that. But so what? You have a model, you can describe the data. So the main thing, as I mentioned before, is generating a prediction, right? So from the model, we predicted that if we stop the immune response, it should decay at a slower rate. The viral titer should go down at a slower rate. So then Stefan, he could do that experiment. He could take this cotton rats and deplete the CD8 T cell, which would mediate this response. And indeed, when he did that experiment, it really matched our model prediction. So there was no fitting here for the predictions. So we got the parameters from the wild type or the for the cotton rats that were not CD8 depleted. And then when he depleted, we got this behavior, and we got the same thing in our model. So it indeed captures the prediction. What it tells us that adaptive immune response is a major controller of the RSV kinetics. And the parameters also told us that the rate at which the cells die because of RSV infection is much longer. So they live for a long time compared to that for flu. So it's a very different infection than flu. And we also found that the innate immunity alone is not sufficient to capture the kinetics. So, but the combination of innate and adaptive immunity is really responsible. So with that, I think I should stop. And as you know, that it takes a village to do this type of problems. And the projects and the people who are involved in this project are marked in bold here. And then we have other projects in the lab, which I did not mention. And our funding comes from NIH. We are really thankful for that. And also from Children's and the Supercomputing Center. So with that, I'd like to thank you for your attention, and I'm happy to take any questions now.
Yeah. Please, if you have any questions, please feel free to ask or clarify your doubts or whatever. Oh, I think there's some questions in the chat window. Yes, Chaitanya, of course, go ahead and ask.
Thank you. Every department has a different convention. Wonderful talk, Jayajit. I'm Chaitanya Afle. I had a quick question about the last part of your talk. Of course, I mean, we're sitting in a pandemic for various reasons. So I guess the obvious question is, at a so-called single cell immune system level, have you contemplated extending this model from the respiratory syncytial virus to the most popular virus in the world at the moment?
No, yeah, not really, but because there are many people who are already doing that. So it turned out that it's a game of speed, right? Because when the virus came about, there hundreds of groups started working in this area, right? So whoever had access to this data, they are much further ahead than us at this point. So we decided not to do that right now.
I understand, but I guess sorry if I may interrupt you, but my question was slightly, I guess I understand that, and we appreciate all of this, I think, in the sense that we all face similar issues. But my question was, if I remember correctly, the RSV is, I think, also an RNA virus, and there are similarities that go beyond the superficial.
Right.
Perhaps there's something one can learn from your model of the RSV at a, in a way, a cellular level, about what might be going on with this. In other words, is there anything perhaps unexpected that one can extrapolate from here without necessarily obviously working on this topic?
Yes, I think so. Yes, that's an excellent question. And so if we have this viral kinetic data for the SARS-CoV-2, I think we would be able to at least explore how the adaptive and innate immunity, what are the relative roles in producing that viral kinetics, and if that can be tweaked to change the kinetics, and then which can probably help with vaccine candidates and drugs. I'm pretty optimistic about it, but we haven't tried it. So it remains as an optimism at this point.
Thank you.
Yeah, thanks.
Anybody else? Please go ahead. If you have any questions, clarify. No? Okay. Okay.
May I ask another question if it's possible?
Go ahead.
Yeah, go ahead.
Make yourself free. Everybody.
So I missed this a little bit, and I think that's partly my fault. But Jadit, when I look at the DNA, the second part of your talk, I think the B-cell stimulation, if I'm not mistaken, with the DNA origami structures, those linear structures, what is their spatial extent, and does that have any implications for the outcome that you see in terms of receptor dynamics?
Okay, so you mean the spatial extent of the DNA rods, right?
Exactly.
Let me see if I can. I do not know the number on top of my head, but they're certainly bigger than 80 nanometers, because that's the distance we. Yeah, so it could be, I think, around 80 nanometers. That should be the length of the DNA. Now, whether increasing that length helps, I don't think we have explored that. But what we have explored, if you make the DNA more flexible, right? Not DNA, but attach something and make it more flexible, or tie those antigens with flexible linkers. So those things have been tested. So if you attach a flexible linker to those ligands, you reduce activation. So it somewhat becomes like
keeping them rigid is important, is it?
Yeah. That rigidity.
And my second question, I'm sorry, I may be asking the obvious. what is the range of spacing going from left to right in this graph? I can make out that rightmost must be around 80 nanometers, but what is the lowest spacing that you can get?
Lowest, I think, is okay. They have 1 nanometer, but they don't get anything for 1 nanometer. So lowest,
Oh, it's
they
7
count
nanometers. I see that
is
now.
7 nanometers.
Yeah, perfect. Thank you. Thank you. Sorry.
Yeah,
You just
thank you.
Hello, may I ask a question?
Yeah, sure. Go ahead.
Yeah, hi Jajit, nice talk. You started this talk with a description of these scale invariants. So in any of these two problems, where and how the scale invariants actually emerge, or you possibly have not discussed that here?
I haven't discussed that, and I don't think we have scale invariance here. So not for this problem.
Okay.
Yeah, there could be scale invariance in other scenarios, but not for these two problems.
Okay. Which kind of scenarios you expect usually these scale invariants that you were talking?
So one problem where you notice scale invariance is where you see formation of biofilms by bacteria. So in the middle layer, I briefly mentioned, you have this bacterial infection. They
Yes.
generate biofilms. And if you look at the structure of the bacterial cells in that biofilm, they can have a scale invariant structure over a length scale.
Okay.
But not in this context.
Okay.
Right. Okay, so basically here in one of the problems you had a more of a lattice model where various particles are hopping around and reacting, and here another one you basically well, you used a series of differential equations. I mean, they were not there was no space dependence. So you basically for this you have various. I mean, what are the other kind of problems that you look at? I mean, within this infection. I mean, I'm more interested in the number of techniques, ways, approaches. I mean, classes of problems. Anything else you want to say?
Yeah. So I for the tools I mentioned, stochastic simulations and solution ODEs, right? But there are types of problems where we use let's say PDEs, and then we can use even field theoretic techniques especially for this for the biofilm problem. One can use field theoretic models to get salient features of that organization. And then I think, and then there are some problems where you solve the master equation analytically. When you reduce the models, you are able to do pen and paper calculations, and then probably some
So you have basically
semi-analytical
the
calculations.
entire range. You have the
Yeah, yeah,
which you
yeah.
okay.
And then now coming back to other techniques. So there's the class of data-driven problems that I did not really mention, and there some of the tools that we use for Ising models or Ising models with disorder. That set of models can be used to develop algorithms and techniques, which I did not really talk about here.
I see. I see.
Yeah.
I mean, I couldn't understand. I mean, how would you use that? But I mean, it's okay. I can ask you, I guess later or whatever. Clarify. Any other questions? Any other general questions or curiosity which anybody else wants to address? Else, I will close the talk. And I'll thank Jayjeet and
Okay. Thank you all. I hope I did not put you to sleep this evening.
no.
So thanks again.
Yeah, so thank you, Jayjeet, for giving us this opportunity to know about your work. Okay, thank you. So I shall end the session.
Sure.
Yeah,
Okay.
bye,
Okay.
and
Bye.
catch up with you.
Bye.